Sequencing a genome involves breaking down the complexity of an entire organism into individual building blocks. These are the A's, T's, G's, and C's that make up the gene sequences within the DNA found in each cell of an organism. It's the specific order and combinations of these four bases that provide the instructions for the development and characteristics of an organism. A reference genome is a genetic blueprint that is representative of all organisms within a species. It's not the sequence of one individual's unique DNA, but rather a mosaic of DNA compiled from multiple donors. This creates a reference point to compare individuals from the same or different species and gives scientists a basis for research into areas such as evolution, disease, and genetic modification. Notable examples of reference genomes include the Human Genome Project and more recently SARS-CoV-2. But to fully understand the application and implications of this concept in modern genetics, let's look at a simple yet staple organism with a surprisingly complex history: beans. Phaseolus vulgaris, the common bean, is an important source of protein and micronutrients worldwide. It's native to the Americas and has particular dietary and economic significance in the developing world. Despite its significance, the common bean genome sequence remained unknown until 2014, when a research team led by Jeremy Schmutz and Philip McLean published their study titled A Reference Genome for Common Bean and Genome-Wide Analysis of Dual Domestication. For the first time, the world was given the opportunity to observe the common bean's importance as a species on a genetic level. Using the reference genome they constructed, Schmutz and McLean identified sets of specific genes and compared these among beans from different geographic locations to decipher its complex history of evolution and domestication. Schmutz and McLean carried out a process called whole genome shotgun sequencing, using common beans from the Andes region of South America. A number of short, sequenced DNA fragments from different positions on the bean chromosomes were aligned into overlapping regions called contigs. The gaps between these contigs were then filled by scaffolding. The result of this was a continuous representation of the DNA of the common bean, containing 587 million bases arranged into 11 individual representative chromosomes. Previous research, particularly in 2012, had established that the common bean existed within two distinct gene pools in two separate geographic locations: Mesoamerica and the Andes. Each pool was derived from a single wild ancestral population thought to be in Mesoamerica thousands of years ago. These two pools then experienced separate domestication events, where they each underwent natural selection and adaptations that modified their characteristics and made them more suitable for agriculture in their environment. Domestication resulted in observable diversity between gene pools, with noticeable differences in plant size, growth habits, and seed size and color. Returning to the 2014 study, Schmutz and McLean sequenced a number of individuals from both the Mesoamerican and Andean populations and aligned these back to their reference genome. This allowed them to screen for genes of traits associated with domestication that have been positively selected over time. They identified 1,835 candidate genes in Mesoamerican beans and 748 from the Andean beans. These included a number of genes associated with cellular pathways involving plant size, including flower and stem size, nitrogen metabolism, and seed size and weight. Overall, they observed that the Andean pool was less genetically diverse than the Mesoamerican, and they were both significantly different to each other, despite both originating from the Mesoamerican wild ancestor. Beyond deciphering the common bean's complex history, what is perhaps the biggest impact of Schmutz and McLean's research was the release of the reference genome to the public. They have enabled a new era of research and discovery by providing a list of candidate genes for genetic modification. These are the genes they identified as being involved in domestication and adaptation, as they have continued to survive selection, making them suitable targets. This has significant implications for crop improvement and food security worldwide. Such genetic engineering is taking place currently, with a study published last year in Canada aiming to improve yield, and in Turkey for disease resistance, among many others. And so, from the example of the common bean, we can see the importance of breaking down the complexity of an entire species to just its genetic blueprint. We now have a greater understanding of the history, structure, and function of organisms on a genetic level, and how we can use these to benefit society, all thanks to the reference genome.